and welcome everyone. Hey, it's Monday afternoon, Labor Day, but we're meeting for class. So the syllabus says exam this week, today, I don't know. Normally what we would do is for the first hour of class, you'd have an exam. And it takes you about 20 minutes, but we'd blow off an entire hour, and there you go. This isn't normal. I don't know. And I'm thinking a couple of things. I'm probably going to put together, if I can figure it out, a Moodle quiz thingamabob that you could take. You'll have a time limit, but you can obviously use whatever resource. Now, if you're smart, what you'll do is you'll make your roommate open it up first. You'll take down all the questions from him, let him blow it, or her, and then you open it up and you get all the right answers. See? And then you can trade off next time. So when I see grades where you made a 45 this time, and then you made an 88 the second time, and your friend made an 88 the first time and you're 45 the second time, well, then we'll know what you're doing. Of course, I may delete this video part and start over again. No, I'll have an exam for you at some point this week. Did you play Lemonade? I hope. And we're going to be playing another lab as well this week. And I'll come up with some sort of thing that you would then, say, yes, I looked at this lab, and these are some thoughts I thoughted. And I don't know, whatever. Hey, how's everybody doing? All right. Now, if you recall, we're working on our macromolecules, and we've gone through the first two. That is, we've done carbohydrates and fats. The monomer for carbohydrates, work along with me now. What's the monomer for carbohydrates? It is a sugar or a monosaccharide. What's the polymer for fats, or the monomer for fats? It is the fatty acid. The polymer for carbohydrates, a good polymer might be amylose. And for fatty acids, a good polymer might be a triglyceride. And an example of a carbohydrate polymer, might be, say, starch or cellulose. There you go. An example of fats might be blubber, or liquid oils. Some interesting characteristics. Carbohydrates and carbohydrate polymers have the ability to form really extensive hydrogen bonding, and that means they can form gels, they can act as thickeners. Fats, they're nonpolar, but there is a class of the fatty acid diglycerides that have that phosphate group on them, and those are amphipathic, and they can form emulsifiers. They can make a stable oil and water mix. That's some stuff. Now, the third group, and potentially the most important group, is the protein class. And proteins. Are you getting enough protein in your diet? Yeah, you probably are, unless you're eating some sort of really weird-ass diet. You probably are. Proteins. The monomer is the amino acid. We're going to talk about that in just a moment. We can put in the polymer. The polymer is known as a protein, also called polypeptide, but that's what it is. And an example of protein is a piece of meat, flesh. That's protein. All right, so now, here's what makes proteins really cool. I'm going to erase and we're going to get into it. So, proteins make up a part of our diet, and they are a critical part of the diet for sure, but they're not like the end-all be-all. You can be on an all-protein diet, and it doesn't make you that much healthier or whatever's. I think that we actually have to really, as a nation, think about how much protein we friggin' eat. We probably eat more protein than we need to, but that's okay. All right, so proteins. To get into it, we have to think a little bit about the assembly of the monomers into a polymer. First off, here's the structure of a protein, and you kind of have to know this one. It is an amino acid, and so it's going to have a carboxylic acid, a central carbon, and an amine group. Let's draw that. So the amine is the nitrogen with a hydrogen. It can be 2 or 3, nobody cares. A central carbon, and then the carboxylic acid. That is the OH, the double bond O. Now, there's going to be a hydrogen on this central carbon. This guy has to have one more bond. It's going to be something called an R group. That is because it is possible for it to be different kinds of things sticking off that central carbon. And it's this way. There are 20 different R groups. Okay? 20 different R groups. And these R groups can be classified into a variety of different kinds of characteristics. Now, the simplest R group is just a hydrogen. That makes the amino acid known as glycine. Now, if you have friends who are taking biochemistry this semester, well, they're having to memorize all 20 amino acid structures. We don't got to do that. It's not too bad, because all you have to do is memorize the 20 R groups, because they all have the same base. But when you see them, you can tell them that, hey, you're also taking biochemistry, and that you got glycine covered. So 20 different R groups. Now, the R groups can be classified into these general sort of categories. They can have a positive charge. They can have a negative charge. They can be, in general, polar. They can be non-polar, kind of fatty-like. They can be acting a little bit like weak acids. They can be acidic, or they can act like weak bases. They can be basic. So this allows for some interesting kinds of molecules now to be built. Now, if we think about this sort of from a larger perspective, when we were looking at fatty acids, they're just a straight chain, long chain carbon. Not a lot of diversity. You might have double bonds, you might not. That's going to change the melting point. But the structure is pretty much just straight chain carbon. With sugars, they're just this massive net negative charge, glucose, 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 glucose. So there's not a lot of interesting information there. It is pretty much homogeneous. Now we have the possibility of building a molecule that actually is not totally homogeneous. It can have a mix of these different R groups. So let's look to see first how we're going to link together two amino acids, and then we're going to see what happens as we start linking together these amino acids. So we're going to draw back up here. So we'll call this one R1. Now, we're going to draw another amino acid over here. And depending on the book and what you're looking at, it can have 2 hydrogens or 3 hydrogens on the nitrogen. Nobody cares. It has to do with the pH of the solution. Double bond O, H, and R2, D2. All right, so now, what we're going to do is link these guys together. It's not all that critical for our discussion, but you'll have the first amino acid. He's line leader, and everybody else stacks up behind him. You can't jump around the front of a large polymer. You just build it from one direction only. But we're going to link these guys together, and it's a condensation reaction. So you're going to look to see how you can make a molecule of water, i.e. pop a hydrogen off here, the OH group off there, make a molecule of water, and bada bing, you have made a carbon-nitrogen bond. This is just a plain old strong covalent bond, and it's given the special name of the peptide bond, but let's not be overly pedantic. It is just a covalent bond. The point being, amino acids are linked together through covalent bonds. Now, when we start linking together these amino acids, because of the R group heterogeneity, the R groups aren't all identical, interesting things can start to happen. Let's look at them, shall we? Yes. Let's think of it like this. We've got a bunch of R groups that we're going to start, or a bunch of amino acids we're going to start linking together. And just remember, the amino acid sequence is dictated by your DNA sequence. That's what your genes are doing, is they're providing the instruction set for individual proteins. So we got a bunch of beads on a string here, okay? Now, each one of these lines represents one of the peptide bonds. Okay, so we know that we can have a whole series of R groups. So let's just put some down here arbitrarily. We're going to put a stretch of negatively charged R groups coming off of this group of amino acids. Some nonpolars coming off here. And now a stretch of positively charged R groups coming off here. Now, when we begin to look at this, some interesting things start to fall out, and that is this. There are multiple levels of structure that can come about from the amino acid sequence. And this is really a critical thing. We want to look at these structures, at the different levels of structure, and see how they're being linked together. We start out with what's called the primary structure, and that's one with a degree, is sort of the abbreviation for primary. So the primary structure is defined as the amino acid sequence. Now, from a molecular biology perspective, I'll tell you, this is really easy to determine because it is simply based on the DNA sequence of a gene. The amino acid sequence is held together by our strongest bond, the covalent bond. That means that you don't break down the primary structure very easily. Stomach acids can begin to do it. There are enzymes that are called proteases that can begin to do it. But for the most part, particularly when you're cooking, unless you set this guy on fire, you're not breaking apart the primary structure. That is sacred. Don't jack with it. I mean, you pretty much are going to ruin your food if you go to try to destroy the primary structure. Now, secondary structure is defined like this. Let's see. I'm trying to think about how much room I got. Sure. We'll try it this way. I'm going to see if this will work. Can you see this? Secondary structure. I really like my fat markers, but yeah, you can see that. All right, secondary structure. It's defined as the interaction of R groups that are neighboring, or another way of saying it is neighboring R group interactions. who are the R groups in your neighborhood? And for the purposes of this biz, neighboring means probably about 5 amino acids on either side. Now, what can happen is like this. You got a stretch of like charges here, and a stretch of like charges over here. Now, we know opposite charges are going to attract, like charges repel each other, and so these guys, they're poking into each other. Their repulsive forces causes the R groups to actually twist around, so they're going to end up looking like this. Every other R group is now upside down. And so you have the R groups now spinning around on opposite sides. And one of the things that happens is this spinning actually creates a helix, what's known as an alpha helix. So we have the nonpolar spot, and then we have another alpha helix. So, neighboring R group interactions, we can get the alpha helix. Another thing you can generate, or you can find in proteins, is that there'll be hydrogen bonding between like every 4th or 5th amino acid that creates a zigzag. And that is known as the beta pleated sheet. And truthfully, this is a bit of a stretch, but it's not too far a stretch, that you can begin to see these kinds of secondary structures in things like silk. It's got a lot of beta pleated sheets, and it actually generates a waviness to it. Curly hair has a certain amino acid sequence to it that is creating these little curls, naturally curly hair. So these are structures that are being generated in a protein in these localized patches, these secondary structures. Now, an important thing for our purposes is what are the bonds that are involved in this, and we can list them like this. The bonds that are involved in secondary structure are typically not covalent. They're going to be things like hydrogen bonds are weak guys. They're going to be polar, non-polar interactions. They're going to be ionic interactions. They're going to be just simple sort of staticky type charge interactions. What they're going to be is really weak bonds. And I'll let you in on a little secret here. In the biochem biz, actually only in my world, we know these as collectively them weak-ass bonds. That is, in Galuski chemistry, how we refer to these things. They are them weak-ass bonds, meaning secondary structure is not all that tough. You look at a protein wrong, the secondary structure can start to break apart. Now, I mean, seriously, hydrogen bonding in this condition, it don't take boiling to break it apart. It might be 110 degrees. You start to run a fever, you start breaking some of these weak-ass bonds. You change the salt concentration just a little bit. You start to change some of the ionic interactions. You change the pH just a teeny bit, and you begin to change these interactions. So these weak-ass bonds, they're delicate little fellas. Now, lastly is the tertiary structure. And I'm trying to figure out if I got room to write. How about this? If I duck walk, I can do it. I was pulling carpet all Saturday, and are my knees stiff? Yes. So if I make groaning noises getting up and down, you'll know why. What would be nice if I could have the floor lower or the board raised, but all right. But for you, I'm keeping it this way. Tertiary structure. That is the next level. Tertiary structure, we define this as non-neighboring. R group interaction. And tertiary structure is maintained also by, you guessed it, them weak ass bonds. Now, tertiary structure means that we've got a protein chain that can fold up. So, in this case, the negative charges over here are going to bend around and interact with the positive charges over here. That's the tertiary structure. So, you got primary structure, amino acid sequence. Easy to predict, it's based on the DNA. Secondary structure is neighboring R group interaction. There are computational models that can do a pretty good job of predicting it, but they're not always exact, or you end up with actually a couple of different kinds of models. Tertiary structure, folding up. Secondary and tertiary, though, weak-ass bonds holding them together. Now, one of the things that pharmaceutical industries are always doing is using computer models to try and figure out tertiary and secondary structure. And it has to do with thermodynamics, which we'll get into a little bit later. But basically, it turns out that for a given amino acid sequence, it might have a half a dozen or so secondary and tertiary structures that it can fold up into. And they're more or less, they're very similar, but they have slight differences, and that difference can be the difference between how they work and not. So it's actually a really cool field, not that you care. But this is the important thing. Primary structure, unless you're setting it on fire, you ain't touching it. Secondary structure, tertiary structure, it doesn't take much. These are weak-ass bonds. You raise the temperature up, you change the salt conditions up or down, you change the pH up or down, you pull some of the water out, you add a little bit too much water, you mess them up. Here is the critical thing about life, the universe, and everything. And truthfully, there are a couple of points that we go, if you got this out of this class, well, heck, you really got something. And this is one of them. That phosphate biz was one of them. Here's another one. It is this. Not that. Now, it is the following axiom or rule set. And you'd think I would just edit that out of the video, but no. This is what it is. All right, you ready for this? This is important. Are you ready? Here it comes. First, protein structure determines protein function. Okay? The way a protein folds up is going to determine how it actually does its job, whether it can do its job and what's the job going to be. The second point is this. Amino acid sequence determines protein structure. So you with me? All right. So a protein has to fold up in an appropriate way in order for it to work. In order for it to fold up in that way, it's got to have the correct amino acid sequence. And lastly is this. DNA sequence determines amino acid sequence. Now let's think about what we have just thrown up here. One thing I'll tell you, that if you could come up with this, certainly in the 50s and maybe even in the 60s and pushing into the 70s, you would probably get the Nobel Prize, and or at least you would begin establishing the modern paradigms behind molecular genetics. But the DNA sequence determines the amino acid sequence of individual proteins. The amino acid sequence is determining the protein structure, and the protein structure determines a protein's function. Consequently, you change the sequence of the DNA, a mutagen. You can change the amino acid sequence. If you change the amino acid sequence, you might just change the protein structure, which in turn might affect the protein's function. This is the mechanism behind cancer. Mutations, very often, if they're going to be deleterious, are changing an amino acid sequence. This is also responsible for why I look like my Uncle Herman. Not because I was standing next to him when I was a child, but because we share DNA sequences that result in amino acid sequences of similar structure and shape, or similar sequence. Now, one of the things is that as human beings on the planet, and heck for that matter, as vertebrates on the planet, we all share phenomenally similar DNA sequence. So the difference between a rat and me, or you, not to pick on rats or me or you, is in fact less than the difference between a rat and a yeast. So we've got a lot of similarity between all vertebrates. We pretty much are all these very similar organisms. And then if you start looking a little bit further in terms of generalities, we all share basically the yeast genome. It's just we have about 3 versions of it, and that's who we are. So life on this planet is actually very, very similar in a lot of different respects. And it's because we all carry out the same biochemical functions, and we use similar proteins, and they have to have the same very similar shapes. Now, that was my molecular genetics paradigm. But functionally, in terms of cooking and food and eating stuff, here's another thing we can think about. And that is this. Proteins are able to work and life is able to exist, or we can call something, it's living, as opposed to, it's dead, because the proteins are able to carry out their different kinds of chemical reactions. Anything you do to disrupt a protein structure is very likely going to disrupt its ability to carry out these chemical reactions, and it will lead fairly quickly to the organism not being able to function, i.e. dead. That was, I'm not sure if the video paused or not. I just wanted you to get that. All right. So, here's the deal. Anything that is going to break apart secondary and tertiary structure, these weak-ass bonds, is going to break apart the structure of the protein. And we have a term here, a protein that has lost its secondary and tertiary structure is said to be denatured. Now, depending on what you're doing to a protein, you can add, like go from an organism that lives normally at 100 degrees Fahrenheit, you and me, raise the temperature up to about 104. You can begin to gently break some of the hydrogen bonds. Very gentle treatment will denature proteins, but it'll probably not denature them so much that they can't renature. And so you can have, if the conditions are very gentle, you can have protein renaturation. It actually can generate back into its appropriate secondary and tertiary structure. But very often, if you are disrupting the secondary and tertiary structure, most proteins are going to end up being permanently denatured. Now, in the food biz, we actually have a couple of interesting examples. Egg white is mostly water and a protein called ovalbumin. Ovalbumin, as a protein, is particularly persnickety to conditions. If you add a little bit of heat, it begins to denature and falls out of solution. You get the white. And egg white is notoriously impossible to renature. Other proteins, though, you can gently open them up, and they will very often renature. Not necessarily will they be functional, but they might have some partial function. But for the most part, when you begin to attack these weak-ass bonds, we denature a protein. And let's look at some of the things that can break apart weak-ass bonds. And let's think about this in these conditions. So, another way of titling this little bit here, this list, is things that you can do that will break weak-ass bonds. So let's go ahead and break them weak-ass bonds. So we can break weak-ass bonds by relatively gentle heat. Certainly, let's say, less than 450 degrees for sure. But the truth is, with a lot of hydrogen bonds, you can probably do it, and I'm sticking in Fahrenheit just for the heck of it, 110 degrees, 120 degrees almost for sure, is going to break apart hydrogen bonds. Now, another thing that can break weak-ass bonds is the salt concentration. It can be too much. It can even be too little. And what this is going to do is start messing around with various polar, non-polar interactions and other kinds of ionic bonds and things. Another thing we can do is adjust the pH. Bring it down, that is, add acid, or bring it up and add base. These are the standard ways you would go about to break weak-ass bonds. Now, another aspect of protein structure and function that I'll just throw in is that sometimes rather than breaking weak-ass bonds, if you chill stuff down, you can allow non-standard hydrogen bonds to form. You can kind of jam stuff up that way too, but usually that's not going to do it. Heating up and more harsh conditions typically are what are going to do it. But let's think about this now in terms of cooking. Pretty much when you're heating stuff up, you're baking, you're cooking your meats and things, unless you're getting a char on there, you're under the levels of moderately gentle heat. And truthfully, 110, 120 degrees, there is a relatively new style of cooking called sous vide cooking, in which you take foods, you put them into a plastic Ziploc bag, and you dunk them in a heat bath that will run at 120 degrees, which I will tell you is warmer than you want to take a bath in, but it's not so hot that you can't put your hand in there. 110 degrees is probably at the high end of what you'd want to take a bath in. But 120, you definitely can stick your hand in that. All right. By holding foods at that temperature for hours, you can begin to break apart the hydrogen bonds. Now, one of the interesting things is you would put food into a sous vide bag and carry it at that temperature, hold it at that temperature for like 2 or 3 days. And you might think, oh God, it's going to be rotten. But the fact is, at 120 degrees, you're denaturing all the proteins in bacteria as well. And so there's nobody to rot. And it becomes effectively cooked. Now, obviously, on the stovetop, you bring something to a boil, you've done that. Boy, you did it in a half. Salt concentration, we can think of salting. Like dehydration, what you're doing is pulling the water out, increasing the salt concentration. You can add a bunch of salt, so like making jerky or corned beef, different things like that. You can brine foods, and that begins to cook stuff. It begins to actually change the bonding and begins to denature proteins. And so salting is a good way to preserve foods. Actually, another way that's not commonly done, but is sugaring. What that's doing is the sugar is pulling the water out, increasing the salt concentration as well. And that is another way of preserving stuff. pH, well, by adding acid, that's like pickling things. So you can preserve stuff by pickling it. You can denature proteins by pickling it. That's one of the things to think about. When you talk about preserving food, it is a denaturation technique. Now, raising the pH up, adding base. About the best I can think of for this is making lutefisk. Anybody ever had lutefisk? I never have. It is a Scandinavian delicacy in which you take fish, soak it in lye, and this is doing a couple of things. It's denaturing the proteins and causing the fats actually to become almost like soap. And then you soak it in a bunch of water and salt, and you wash all the lye out, and you're left with this gelatinous white mass that I can't imagine tastes all that good, but people eat it. It is another preservative technique. Another way of cooking with acid that maybe you've heard of if you've ever been to Mexico, perhaps, is something called ceviche, which is really quite amazing. Ceviche is made... Ceviche. It's made with like absolutely fresh fish. No, just straight off the boat. And you chop it up, and then you begin squeezing in limes or lemon juice, dropping the pH, and adding a little bit of jalapeno peppers, and you have that with corn chips. Oh man, is that good. And you can actually watch the fish become opaque and start to turn white like fish that has been cooked would look. And that's because the acid is denaturing the proteins. So when we start thinking about cooking is basically protein denaturation. Pretty much anything that we would do to denature proteins is, in fact, a form of cooking. And another aspect to this is if you want to keep stuff from growing, either on your food or rotting, your food kind of decomposing, what you do is you denature all the proteins within that material. Now, if you want to keep it sterile or keep it from getting bacteria, you got to seal it up first, put it into a jar, into a bag, and then you heat it up. Bada bing. There may be bacteria and stuff in there, but all the proteins are denatured, all the protein in your food's denatured, everything's denatured. You keep it sealed up, it's preserved. It's good for a very long time. And so this is basically the biochemistry behind putting away foods, canning and things. So you got to make sure that you do get the heat all the way through, or that you have a low enough pH that the food is going to be sufficiently acidic. Otherwise, you may not be denaturing everything. All right, so we now have our 3 classes of macromolecules: carbs, fats, proteins. Now, in true biochemistry, the 4th class is the nucleic acid class, but we're not going to worry about that. Our next set of discussions is going to be talking about how we can begin linking these different molecules up and actually breaking them apart and building stuff. The idea of metabolism. And it's a very cool, little bit complicated story, but we'll be telling it in the form of a grand opera, or at least a good musical or a play. And that's what we'll be getting into. So, okay. you had to sit through this entire lecture, but hopefully you did. And here's my thought. I'm going to put together a quizzy type exam-y thing this week that'll be covering this first chunk up to here. And I'm going to drop, probably tomorrow or Wednesday, another discussion lecture thing on starting the next set of things. That's going to be for the next exam. But the exam is probably going to be A, really, really simple. B, graded pretty easily. C, I'm hoping to make it into a timed like Moodle quiz. So at least you got to do a little bit. But don't worry about it too much, please. Read through your notes, learn some of the stories, talk to your friends. Tell your friends these stories. See, and you can be a smarty pants. Thanks for your attention. Rock on. I will see you cats later.